Morning, everyone. Hope you're doing well this morning. I decided to go to Italy over the weekend. Here I am at the Leaning Tower of Pisa. Hope you had nice weekends yourself and enjoying the beautiful weather. Hopefully it's not too hot where you are. Let's see. What do we want to talk about? Well, we do have an exam coming up. I guess we should talk about that. It's at the end of the week. Let's go ahead and look at that information, and then we can chat about it a little bit if you'd like. Let's see here. Oop. Let me just redo that. Sorry. Make sure all the sound is on. You'd think after years of doing this now, I do it perfectly every time, but there's still some things I forget to click. All right, there we go. Oh, shoot, cancel. Okay, I think it's working fine. Let's hope. It's doing some weird stuff again, but hopefully it's okay. So hopefully you're seeing the PowerPoint slide on your screen about the midterm exam. It can be taken any period, any 80-minute period between, I guess, Friday at midnight, Friday at 12 a.m., all the way through Sunday at midnight. So Friday or any time Saturday or Sunday. It is an open book test. Okay, you can use any notes, any video, anything like that. And I should warn you, though, and this is something that happened in the past, people think because it is an open book test that they don't need to study, but trust me, you will not have time to look up every question. There are probably 60 questions or so. I haven't finished the exam, but I can tell you more about the specific number of questions probably by Thursday. It's going to be about 60. You have 80 minutes. That's a little bit over a minute per question. And I can promise you without studying, there's no way you'd be able to pass. So it's important you study. But if there's some questions that you have problems with, you can go back and look them up. That's not a problem. So it's open book. This is, part of the quarantine format that a lot of us professors are doing. But that doesn't mean you can cheat. And what I mean by cheating in this context, actually, it's two things I want to mention. One, of course, is that it must be taken alone. We don't want you on Zoom with another person in the class trying to work the questions out. That's just plain cheating. And the other thing is, I'm going to restrict the test so it has to be taken within the 80-minute period. We did have a few folks last year who pretended they lost their internet connection so they could look up questions. It doesn't make a difference now. If you do lose your internet connection, hopefully you'll be on a good internet connection. Everything will be ready to go. If your internet connection is a little fragile, you might want to go somewhere where it is less fragile, because you will have that 80-minute period to take the test. And if you do get kicked off, you're going to have to go back on right away and finish it within that 80-minute period. In other words, the 80 minutes continues to be counted. The format of the way I'm setting up the test, the format will not allow you to take a break and then go and stop the 80-minute period, go look up answers and come back. Okay, so it is an 80-minute period, even if you do lose your connection. Okay, but again, it should be so straightforward. In fact, with this format, people have been doing very, very well on these tests, so it shouldn't be a problem for you. It covers everything through at least much of Thursday's lecture. Today we should get through smell, and then we'll start taste and then finish taste on Thursday. If in fact we do have a chance, we might just start the touch perception material. I will let you know that when we do that, and that will not be material on your midterm, that will be material tested on your final. So everything's through the taste content material, which also involves chapter 5 in the book, and the discussion section material through yesterday's discussion section. Dylan will be presenting new material, I guess, on touch in Wednesday's class, and will also give you a chance to ask some questions for the exam during Wednesday's class. So there will be a little bit of a review session, but it is guided by your questions. He's not going to review the entire class up to this point. He is there to answer your questions, and he also has an office hour or two. You can take advantage of that to ask questions as well. One other thing about the book material, it's important that you read through the preface. There will be some questions on the exam that are derived right from the preface material. So don't forget to take a quick look at that section as well as everything through chapter 5. Okay, let me take a little break from this slide and see if you folks have some questions. Right. Any questions that I can help you with regarding the midterm exam that's coming up? You either can say it out loud or discussion, or I'm sorry, chat. Use the chat function. Anything? Okay. All right. Hopefully you're feeling okay. How many participants we got today? Okay, that's not a lot. That's all right. Hopefully the people that are doing this asynchronously will be okay. I always worried a little bit about them, but it's up to them. Okay. All right. How can we access? Oh, Hector, if you go back to the iLearn page first of all, I posted the link there. Those lectures are on YouTube. We have a dedicated YouTube channel, and so look at your iLearn page for the class, and there'll be a link there for the YouTube page where we post the lectures. Okay, what chapters, pages from the book will be good for study prep? Well, it's everything from the preface through chapter 5. Yeah, go through all that, and then you know how the test is derived. We're going to be deriving a lot of questions from the lecture, and you know some of those questions. You're going to know about, I don't know, let me think about this, about, I don't know, a little less than a quarter of the questions. You're going to know about 20, 25 questions already from the practice questions we've been doing in class, so that helps. But then a lot of the other questions are derived right from lecture material, and you can use the book to help you understand the lecture material. And then we do derive questions from the discussion section that was not covered either in the book or in the lecture. So Dylan is presenting some material in those discussion sections that is new, and that will be tested as well. And then there will be questions derived from the book material that is not covered either in lecture or discussion section as well. So it's, you have 3 sources. Fortunately for you, there's tons of overlap between those 3 sources, so it shouldn't be overwhelming amounts of material. Okay. Other questions that I can help you with on the test? All right. I assume you folks have taken these online tests before since we've been in quarantine. It works pretty well. Rarely do we actually have technical problems at this point. In the beginning, it wasn't so smooth. You may have taken classes where the online tests were monitored. I'm not doing that. You know, nobody's going to watch you take the test. There's no Zoom proctoring or anything like that. That being said, you know, it's important that you not take the test together or anything like that. And the clock continues to tick even if you do lose your connection. Make sure you have a nice connection. Okay. Anything else? All right. Then let's continue with our material for taste. I'm sorry, for smell. Yeah, we haven't finished some of my favorite smell material, my favorite stinky stuff. All right. Cool. All right, so let me just set some, oh. All right, shoot, I forgot to share sound again. I got to do that again, sorry. Why it doesn't just default, I have no idea. No. Let's try this again. Okay, I am sharing. All right. You have no idea what's. I mean, it's just I'm getting all these warnings that the sound isn't sharing. We'll find out if the sound isn't sharing. I think it says it's not sharing, but it actually is. That's what we found out from last time. Okay. Anyway, let's continue talking about smell as we had left it last class, and let me remind you of where we were. What we were talking about now are the unconscious aspects of smell. And we left it here with Martha McClintock. Remember her? So these are, let's take a step back and get the big picture here. We've been talking about smell, and we started talking about smell in the context of first of all, what we could consciously detect if we're asked to, and as it turns out, we can do things like recognize our siblings and our family members and our romantic partners. But there does seem to be this genetic component that gives us an advantage to recognizing people that we have some genetic connection with. And we talked about other aspects, as did Dylan in his sections with regard to scent tracking and those sorts of things. Okay, hold on just a second. I apologize. Let me see what's going on here. Oh my gosh. I can't see. Oh, I apologize. Where's my video? Sorry about this. Why is this? I can't, hold on just a second. Oh my gosh. All right, sorry, I can't see my own video, which isn't good. I want to make sure that's recorded so you guys get to see that as well. Everything seems to be on. Let's try it one more time. All right, man, oh man, see, this is why we have to go slower. Yeah, I have no idea why this isn't working. All right, I assume my face is on there somewhere. I can't see it right now. I have no idea what's going on. I'm really sorry. Are you guys able to see my face? Just give me a thumbs up or something like that. Or actually,
yeah, we can see you.
You can see me? Okay. I can't see me, but hopefully it's working okay. I have no idea why I'm having problems today. Anyway, okay, so let's go back to the material. I apologize. So Martha McClintock, and what she was able to show us is one of the things that we can do unconsciously with smell. In addition to having these influences of things like a fearful odor from another person who's scared, as well as things like a clean odor that we do not detect. These are all things that are subliminal, meaning below our level of conscious awareness. It does have subtle but systematic effects on us. And this is one of the reasons that smell has been kind of an intimidating thing to people, and that these things don't have the sort of conscious access that context of visual information or auditory information gives us. And for this reason, people are, kind of a little put off by smell and wonder whether or not we actually can be manipulated, coerced to do things. And the big picture here is, no, we cannot be coerced to do things we don't want to do. That being said, there are subtle influences that are worth understanding, because the brain is processing odor information long after we are adapted to it consciously. So when we no longer are aware of a smell, our brain continues to process it without our awareness. And one of the things that seems to occur with is the kind of odor that women give off to each other to entrain menstrual cycles. And that's where we left Martha McClintock, menstrual entrainment, where she found people would entrain that were living together. However, this was really controversial when it came out, because she wondered whether or not this was occurring in the same way it occurs for mice and a number of other mammal species, where animals that are cohabitating seem to entrain their menstrual cycles. People were really kind of put off by this argument, though. They thought, well, yeah, maybe mice and deer and things like that can show these sorts of entrainment patterns, but people are much more complex and won't show that. And if they do, it's not because of pheromones. In fact, I think there's a lot of people still who don't believe that humans have any sort of a pheromone influence at all. We left this off at the kind of a criticism of the different types of methods that Martha McClintock used regarding self-reports, and the fact what proximity means here is that she found that that roommates weren't necessarily the ones that entrained most strictly. Sometimes it was friends that lived down the hall that would seem to entrain more strictly. And that seems a little surprising given that the proximity of a roommate and the exposure to a roommate in terms of the amount of time is going to be longer. Okay, so she comes back and does a very, very clever experiment, which I think is very compelling evidence that this does have something to do with, the chemical signals that people can give off to each other. And what she did is she took sweat samples from ovulating and pre-ovulating women. Okay, so it's a little bit of a complicated experiment, but I think you'll get the gist of it if we go through it slowly. Okay, so she first brought in a bunch of women. This is no longer in her dorm. This is now when she's a graduate student. I don't remember where she went, MIT maybe or something, someplace like that. She decided to really kind of study this very carefully. So she brought in a group of undergraduates and first just took sweat samples from that and determined when they were during their cycle. And so what she then took the samples that were from ovulating women and pre-ovulating women, and then use those samples as stimuli to a new group of women who she was going to test to see if their cycles would shift at all. And it was one of those experiments where the receiving women, the women that were presented these sweat samples, had no idea that they were being presented sweat samples. In fact, these were presented to the upper lip of women, but the women were just told that they were being presented different types of, I guess it was an alcohol concentration, like rubbing alcohol. And that's really all they could smell, what rubbing alcohol smells like, right? The thing about the rubbing alcohol is it was kind of used to mask any sort of possibility that the women would actually smell any odor from these donor women that the pre-ovulation and ovulation sweat were taken from. Okay, so they're applied to the upper lips of women. And then I guess that was done, every day for a few weeks. And the question was whether applying the ovulation versus pre-ovulation sweat to these new, receiver women would have any influence over their menstrual cycles. Okay, so again, they are getting a direct presentation of the sweat samples, okay, to, in this case, their smell system. And the question is, without them being, aware of what the experiment is like, without them being aware of that they are getting the ovulation and pre-ovulation sweat, does it have an influence over their cycles? And the answer is absolutely. In fact, depending on whether the receiver women, the women that got the sweat on their lips, okay, got the pre-ovulation or ovulation samples, okay, their cycles would shift 2 to 3 days earlier or later. And I can't remember what it was. I guess if they got the ovulation sweat, then their cycles would actually speed up 2 to 3 days. If they got the pre-ovulation sweat, their cycles would actually delay. Maybe it was the other way around. It actually doesn't make that much of a difference. You'll never be asked the difference in that, says in the book either way. But the point is that the cycle did change. I guess if they got the pre-ovulation sweat, they sped up a little bit, and they got the ovulation sweat, they actually delayed a little bit. I think that makes more sense. But I don't care too much about those details. They were shifted in systematic ways, which I think is pretty strong evidence that it does have something to do with our smell system, and it does have something to do with the chemical signals that the women are giving off through their sweat. Now, the idea that you can shift menstrual cycles using a chemical signal that is detected by the smell system in other animals is called a pheromone influence. But even when she presented these results, okay, a lot of her fellow researchers thought, I still don't think this is pheromones, because pheromones with animals work in a very, very different way. Okay, yeah, it's true that it is probably pheromones, in mice that do synchronize them. So this is sometimes called a primer pheromone. We're going to see that there's different classes of pheromones here. The primer pheromone would be the type of pheromone that does sync mice when it comes to their menstrual cycles. Okay, so it does entrain a mouse menstrual cycle. That's considered a pheromone influence. And yeah, Martha McClintock's able to show something in women that do the same thing, but there's still lots of other types of pheromone influences besides this pheromone influence, this primer pheromone influence, that seems to act very, very differently in animals. So besides the primer pheromone that seems to change the metabolism kind of more subtly with these animals, okay, there is something called a releaser pheromone. And we know the biologists, the zoologists know a lot about these sort of pheromones as they occur in animals. We talked a little bit about one of these pheromones, and this is the pheromone that causes ants to scatter when they smell that another ant has been threatened, right? Same thing happens with mice. These seem to be chemical influences that work through the smell system that are much more dramatic. It's like the animals have this like reflexive sort of behavior where they smell another animal and they run for the hills. Remember, we talked about this. Like, this seems like a much more dramatic behavior than the kind of more subtle behaviors we saw with the fear smell and humans, right? So that's one, the ant scattering. Another very famous one is the pig mating stance, that when a female pig is an estrus, when she's going through her fertile cycle, if she is exposed to a male pig, okay, the smell of a male pig, right, then she will kind of reflexively go into a mating stance in order to procreate with the male, right, in order to have the sex in order for them to have offspring. So she will kind of spontaneously go into the female pig mating stance so the male can mount her and everything. So that seems reflexive too. Okay, so those are the types of pheromones that have long been studied in animals. But we don't see anything like a releaser pheromone, it would seem, in people, right? Even if we do show these subtle effects of fear, right, with placing the sweat of a scared person on the lip of somebody who's in an experiment, and their eye blink becomes a little bit greater, right? They become a little bit more vigilant on certain tasks. Remember this, right? They're much more subtle. They are not, they don't seem like big reflexive sort of behaviors. You can't present somebody a fearful order and then have them run out of the room. It doesn't work like that. For that type of reason, people say, well, I don't think these are real pheromones, Martha. They do not. It does not induce people to act like animals, right? Because humans don't really seem to have much of at least a releaser pheromone. Maybe they show a primer pheromone a little bit, but I doubt it's going to be as consistent as the mice menstrual entrainment anyway. So let's think about that. So what would be the primer pheromone in humans? Well, the menstrual entrainment, certainly. There's also some evidence with menstrual cycle shifts that are induced by the proximity of a man So maybe you've heard this before, maybe you've experienced before. When a woman cohabitates with a man, very often their menstrual cycles will become more regular. So maybe that's something like an example of a primer pheromone influence, but people were still very, very suspicious of it. People, were thought that this is still not as strict. It's still not as kind of hardwired as what goes on with mice, where and it's true, there seems to be less variability with mice entrainment, fertility entrainment, than it seems to work with people. Okay. And certainly when it comes to that releaser pheromone, the one that shows kind of a reflexive behavior, we're not seeing anything like that, right? So if a woman's is having her fertile portion of her menstrual cycle, and she smells a man nearby, is she going to go into a mating stance? Absolutely not. She might not even, have interest in dating the person or anything. She might be very far from anything romantically inclined when it comes to that portion of her cycle. Okay, so it is not like this dramatic reflexive behavior that we see in pigs and many other mammals. And the fear thing, again, we've said this a number of times already, that when you are exposed to the smell of a fearful person, you are not going to be, sorry, I've got a little, get rid of this. You are not going to have a reflexive sort of behavior in running out of the room. Okay, you might show subtle things, but not that sort of dramatic reflexive behavior. The only time that you might see something a little more like a release of pheromone, and it's very unclear if this should count, but it is fairly systematic. When an infant, very young infant, as you can see in that picture down here, when a very young infant is, sorry, is this working? Yeah, there he is. Hopefully you're seeing this. Yeah, there he is. When a very young infant is exposed to his mother's smell, and that's what you are seeing here, a little pad with his mother's smell on it, he will start kind of wriggling around and trying to move towards it. And it's a pretty systematic thing. Now, these guys move very, very slowly when they're this young. It takes them a long time to get over there, but they kind of will orient towards it and start wriggling towards it. And that seems to be, it's hard to know what's going through the infant's little mind then, but it does seem to be something like automatic behavior. It seems somewhat reflexive. But again, it's really hard to tell. And it's not the sort of reflexive behaviors you see with animals that are exposed to a pheromone that's related to menstrual cycle or a pheromone that's related to fear. And what folks like Martha McClintock have done is said, okay, that's fine. She agreed that, we're not seeing that sort of reflexive behavior, but maybe that doesn't mean that it's not a pheromone influence. Maybe it means that the type of pheromone influences that we've been categorizing for animals is too restrictive. Maybe, because animals can't report to us what their experience is. We don't know about all the potential subtleties that might happen with pheromones with animals. Maybe we need to broaden our understanding of what counts as a pheromone. So what she says, along with her colleagues, is that the idea of these releaser and primer pheromones being the only classes of possible pheromones is too restrictive. That when it comes to, understanding people, we can kind of broaden what we would consider a pheromone. And in addition to that, it could be that by broadening our categories of pheromones, we actually might understand more of what's going on with animals as well. Okay. So what does she propose? She proposes something like this, that besides releaser and primer, which you might not see a lot of in human behavior, there are other types of pheromone influences. For example, a signaler pheromone. Okay, what does that mean? Well, it's going to be a pheromone. Okay, again, kind of a chemical, messenger in the secretions of sweat of an individual that conveys information to another individual through their smell system about, kin, like genetic relationship, gender, and reproductive potential. So, in other words, it could be that, when we are seeing these influences we talked about last class with regard to the t-shirt experiments, when people are trying to recognize their siblings and, recognize their romantic partner and, that type of thing, that's also a pheromone sort of signal, but it kind of provides information. It doesn't necessarily induce any sort of reflexive behavior. It acts as a signal. Therefore, the name signaler pheromone. So we're going to see some kind of interesting examples of this in just a second. And she says, let's consider another type of pheromone, one that doesn't, release a reflexive behavior, but one that has subtle influences. Okay, I'm sorry, I forgot to put that up. The t-shirt tests are what I was talking about with regards to the signaler pheromones. This other class of pheromones she's proposing, though, she's going to call modulator pheromones. And these are pheromones that might not induce a reflexive behavior like the scattering of the ants or the mating stance of the pig, but has subtle influences on mood and emotion. And through testing humans and getting all the subtle sorts of responses that human experiments can give us, we can learn about that type of pheromone as well. And so we've already seen one, that fear-inducing odor, right? So we can, ask humans to respond to, scared faces, or, I guess, what else did we talk about with regard to fear? The vigilance, being more kind of heightened when they are being exposed to a scared smell. Because we can do that with human behavior, with human tests, unlike animal tests, we can find these subtleties, okay? We can find these subtleties. And so these other pheromones might exist for all species, but humans allow us to really kind of get in and find, the type of subtleties that pheromones might induce that animals don't, okay? So let's go ahead now and look at other types of what might be signaler or modulator type pheromones in humans to see if we can kind of continue this idea that, in fact, humans are to some degree, they are influenced by pheromones. Okay, so let's look at some possible signaler types. And this is something we discussed briefly when we were talking about some of these t-shirt tests. Now we're going to get into some of the details of it. We talked about this idea of MHC compatibility, the major histocompatibility complex. Now remember that has to do with the scent signature, that we all have a different smell unless we have identical twins, and that's because of our genetic code, and that thought that genetic coding might occur in this part of the genome, the major histocompatibility complex, that not only determines our scent signature, but helps determine our immune and autoimmune systems. And what happens with people smelling t-shirts, especially women smelling men's t-shirts, is they tend to like the smell of a man's t-shirt who has, again, this complementarity in their MHC, okay? Why? Well, because complementary immune systems allow for stronger offspring, okay, right? So if I, in fact, procreate with a woman who has an immune system that kind of complements mine, and what that means here is that it kind of fills in the hole. Say I don't have good immunity to a certain type of virus. If my wife does have immunity to that virus, and I provide immunity that she doesn't provide in her major histocompatibility complex, if we can fill in those spaces, those gaps in each of our own immune systems, we're going to have kids that are really, really strong in terms of their immunity. And so potentially, if this can be signaled, if people's scent signatures can convey something about the kind of general form of their potential genetic immunity, then we can pick mates that will allow us to have strong offspring in terms of their immune systems. Does that make sense? That's what complementary means here. Well, we do know that this works with animals. So mice tend to, procreate with other mice that have this complementary immune system. We know the same thing is true of birds, fish, and lizards. Is it true in people? Well, so in other words, should we look at individuals' immune systems that are married to each other and look for this complementarity? Okay. And the answer, certainly research has been conducted on this, and I hinted to it before. There's not a whole lot of evidence that people are completely guided by the complementarity of a spouse's immune system. People are more complex. Okay. So again, remember I told you there are websites you can go to and you provide a little saliva, and then you take a saliva from your significant other and you have it analyzed to see whether or not you've got this kind of complementarity between immune systems so that your offspring is going to be healthy. Yes, that service is provided. You can get results back that are based on the DNA test. However, we do know that spousal choices, by looking at married couples, there's a tiny bit of evidence that might play a role, but so many other things play a role in spouse selection, right? Intellectual commonality, attractiveness, visual attractiveness, compatibility, cultural things, all that sort of stuff comes into play. So you don't see a lot of evidence, like you do for mice, birds, fish, and lizards, that people will choose spouses based on the compatibility between MHC systems. But you do see some interesting research. So attractiveness, we've said this before. Odor attractiveness, when you ask women to smell t-shirts of strangers that are men, you give them 10 t-shirts, they will often pick the smell of the t-shirt from the man who does have the most MHC compatibility, complementarity to themselves. Okay, that is something we've mentioned a number of times. All right. And there is some other evidence that I think you're going to find interesting that suggests that it still might play a subtle role in things like mate selection for humans as well. And this is always something that people find very interesting. This was done with undergraduates who were in relationships. They did, in fact, what these companies are, provide. They had the, man and woman come in that are in couples and provide DNA samples, and then looked at the complementarity between the MHC systems. And then they asked questions about their sexual life, their fantasy life, that sort of thing. And they found this, that women were more likely to have extra relationship affairs. These are undergraduates in relationships. They're more likely to have extra relationship affairs and fantasies if there is less compatibility, between their MHC and the MHC of their partner. All right. So if the MHC of their partner is not complementing their own, they're in a relationship with them, certainly, but they do show that they're more likely to cheat and that they're more likely to have fantasies about other people if the MHC doesn't have that kind of good complementarity that the offspring would benefit from. Okay, so that doesn't mean they're going to necessarily, not get married to that person someday, but at least at that time, when you're in college, when you're in that age range, it does seem to have some bearing on extra relationship affairs and fantasies. If there's not that good complementarity between the MHC systems of people in the relationship, then women are more likely to have extra relationship affairs and fantasies. That likelihood increases during her ovulation. Okay, so these folks were asked these questions at various times during the month, and they found that women would report having greater likelihood of sexual fantasies about other people if their MHC wasn't very compatible with their partners. That increased during the ovulation cycle, ovulation portion of her menstrual cycle. Okay, so ovulation tends to amplify the influence of MHC compatibility on her fantasies about other people and possible relationships with other people. What do we make of this? Well, it suggests that, yeah, the MHC compatibility does have some subtle bearing. It might not mean that these two folks aren't going to get married and have a wonderful, romantic life forever, but at least at this age, you do see this sort of influence. And it probably is based on subliminal odor, at least to some degree, right? So that's how this sort of thing is conveyed. MHC is conveyed through odor. We know this. So that, in fact, could be one of the things that would be classified as a singular pheromone. Now, again, let's make sure we see it as a singular pheromone. We're not seeing this as a releaser pheromone. The woman is, when she's going through her ovulation, she's not all of a sudden breaking up with the guy or something like that because it's the MHC isn't compatible, right? She might be likely to have more fantasies about other people or something like that, but it is not the reflexive behavior you see with the pig's mating stance, with the ants running away, all that sort of stuff. These are subtle sort of influences. So Martha McClintock says, let's not call these releaser. Okay, let's call these signaler pheromones that kind of provide information but don't induce reflexive-like behavior. Okay, that's one thing. What else? I think you'll find this really interesting. Another type of signaler pheromone. We're still on this topic. Another type of signaler pheromone, and this has to do with symmetry. And when you kind of look at the biology of what's provided in pheromones like these type of things, you learn a lot about symmetry. It turns out to be that visible symmetry in a person's body, visible symmetry in a person's face is predictive of some pretty interesting things. Okay. So what do I mean by symmetry? First of all, I want to assure you that you all have symmetric faces and bodies, in terms of being within the normal range of things, most likely. But animals sometimes show less of a symmetry. So if you kind of just draw a line down the middle of an animal or a person's body, and you look at the left side of their body and the right side of their body, you might see differences in things like, horse's hooves or maybe other aspects of the leg, or very often you do see it in the face to some degree. And the same to some degree does hold for faces. Now, it's interesting. If you go to the internet and look up asymmetric faces, you can see, people that have had accidents or something like that, and their face looks asymmetric because of that. But that's not what we're talking about. We're talking about asymmetric appearance of a face that's based on genetics. And what pops up most are images of suspect lineups. And that's exactly where I got this poor individual's face right here. It's from the suspect lineup. And you find that a lot of times there's a higher incidence of asymmetric looking faces in suspects or perpetrators of crimes, which is interesting because it kind of does follow a little bit of what we know of animals. So in animals, we know this, that lower body symmetry, meaning more asymmetric appearing animals, are known to have slower growth and reduce longevity and reduce fertility. Okay. In other words, it probably does signal something about a kind of lack of good genetic health. So they grow slower, they don't live as long, and they have less offspring, they're less fertile. Okay, so it might have something to do with genetic health. So if you're an animal looking for a mate, whether you're conscious of this or not, you know, it makes sense for you to want to mate with a symmetric animal. Okay, I don't know what goes on in the minds of animals when they're looking for mates, but that is something that you know, you find animals, you know, kind of wanting to mate more with animals that have this, you know, symmetry. That's at least visible, you know, to the scientists, as it turns out. What about humans? Okay, so yes, there are these instances of asymmetry, and you can see it's pretty dramatic asymmetry, because I know everyone's going to look in the mirror after class is over and say, geez, you know, my face does look a little, everyone's face is a little bit different, but probably not as different as this guy's. So this does happen in humans as well. And what does that mean? Well, you find that people that have lower body or face symmetry in humans do show a little disadvantage in some areas, right? So they do have lower IQs to some degree and worse physical and mental health. Now, we're looking at big trends. Again, first of all, we're talking about pretty major asymmetries here. It has to be an asymmetry that's based on genetics. And, you know, not like everybody that has an asymmetric, genetically based asymmetric face or body is going to have a lower IQ and worse physical and mental health. We're talking about kind of big, okay, yeah. Sorry, I'm just getting notes on my screen. We're talking about big data here. We're talking about lots and lots of data. And so what you find is that, in fact, you know, if you look at the big data, you look at average, yes, they do have worse physical and mental health, which might help account for why you see a disproportionate number of asymmetric appearing faces in lineups, in criminal lineups. Okay, that would make some sense. All right. On the other hand, people that show high body symmetry and facial symmetry as well do tend to be those that have more sexual partners. And this amazing, men that have higher body symmetry tend to be able to induce more copulatory orgasms in women. Okay, I was very surprised to see that, but that is something that does show up. All right. All right, so that's something you just learned about visual symmetry. We will talk about that later on when we talk about multisensory perception, because you're going to see this also is something that seems to be conveyed through smell. Therefore, the title of this slide, you know, the smell of symmetry. Because if you take pictures of guys and measure their symmetry as they come in when they're in the lab, and then provide T-shirts, conveying their own smell, and ask women now to smell those T-shirts and find the ones they find most attractive. Yes, they will partly base that on the complementarity, but they will also choose T-shirts that are from men that are more likely to be symmetric in their appearance. Let me say that again. You get okay, you're a female subject now. You get 10 T-shirts presented to you. You say, okay, order this in your most attractive smelling T-shirt to the least attractive smelling T-shirt. You're told it's from men. So which male smell do you like the most? Which male smell do you like the least? And what you find is, yeah, that the woman might choose the most appealing one from a man that has that complementary MHC. But beyond that, the T-shirts that are in the group of smelling appealing, smelling nice, are more likely going to be from very symmetric appearing men, and versus the T-shirts that, no, these aren't appealing to me at all. These are from T-shirts of men that are less symmetric. Okay, so what are you smelling here? Well, to say that you're smelling symmetry is kind of not quite right, because what you're smelling And what you're seeing, okay, with symmetry is probably genetic health. So to some degree, not only are you able to smell the MHC compatibility with another person, you're smelling their genetic health, which is being reflected not just in their sense signature, but in their symmetry, in their visible symmetry, which is interesting. So it looks like, genetic health is something that can be conveyed visibly through the appearance of symmetry, okay, as well as through the smell system. Okay, as an appealing smell that's reflecting something about the MHC, structure of that person. Both of those things, the body symmetry, the facial symmetry, and the appeal of the smell is being determined by genetic health. So you can get that information both ways. It's potentially a great example of the signaler pheromone that Martha McClintock and her colleagues are suggesting we have, right? And again, if a woman is in her ovulatory period, this is more likely to occur. So, oh, yes. So it not only does ovulation heighten a woman's sensitivity to this, but fertile phase women, okay, ovulation also like the smell of dominant men better. So how does that test it? Okay, so we're going beyond now this symmetry thing. This is kind of another topic to some degree. What you find is, so let's do it a different way. We're gonna do this experiment a different way. You bring all these men in, and now you're not going to measure their symmetry. You're just going to have them provide donor t-shirts, and you're going to do that personality test. And one of the things you can get from a personality test is whether a man is kind of more like an alpha male or beta male or whatever those things mean these days. Whether the person has a lot of motivation, whether they tend to be a leader, that sort of thing. And for whatever reason, you find that women, especially during ovulation, okay, will tend to like the smell of one of these dominant men better than the less dominant men, as based on the personality questionnaire, right? Which is interesting. Again, let's step back and show and remind ourselves that we are not talking about determinate behavior. It's not like, when a woman's in her fertile phase, she's going to run to the most dominant men and say, let's have babies. It doesn't work like that, right? Women, we are married or in relationships for long periods of time. It's not like we swap like that. Most of us as humans, right? That quickly. However, you do find these kind of subtle effects of attractiveness of smells based on different portions of a woman's cycle, and all these other characteristics such as symmetry, and which is going to reflect genetic health, and to some degree dominance, which might have a genetic component to some degree as well. Okay, interesting. Again, though, these are not releaser pheromones. These are signaler pheromones. They don't induce reflexive behavior in people. What about men smelling women? And this is, there's less kind of very consistent sets of findings like this. So you've probably gotten the idea that a majority of this research, at least when I was finishing the book, the majority of the research had been done on heterosexual women smelling heterosexual men's t-shirts. Okay, there is some research, more recent research on same-sex couples, but the majority of it involves women smelling men. There is research also with men smelling women, but the results, as I said, are less consistent. But one that is consistent and worth conveying is that men are asked in experiments to smell the t-shirts from women that have been worn by the women at different points in her menstrual cycle. Okay. And what you find is that men like the smell of a woman's t-shirt when that t-shirt had been worn during the woman's fertile cycle, during ovulation, better, okay, than before ovulation or during menstruation or something like that. There is evidence, in other words, that men like the smell of a woman better when it's during her fertile time. Again, it's not a reflexive behavior. These are signaler pheromones, but it is interesting that, in these T-shirt experiments, that's what the pattern of results will show. And so in a way, it's interesting because forever, the biology research, the anthropology research suggested that like a few other mammals, not too many others, but a few other mammals, that females in the human species hide their fertility. Okay, they don't have, the change of appearance that, certain other primates will. Where their sexual organs, change color or get swollen or anything like that. We've long thought that humans are kind of hide their fertile period, where other animals signal their fertile period, right? But here there does seem to be some evidence through, through smell that maybe there is some signaling of fertility going on. Does this have a reflexive influence? No, again, it doesn't. And I'll be emphasizing this again and again. A lot more goes into it than just smell. But smell might play a subtle role through acting as a signaler pheromone. Okay. All right. Cool. Good time to take a break and do a question. And this cool stuff, I think, is so interesting. Oops. What just happened here? There we go. I will stop sharing. There I am. No idea why this is working like this. Okay, let's do one of our questions. Let's see. We talked about anosmia, infants and smell. Okay, so this goes back to last week's lecture, or last Thursday's lecture. Which is true about newborn infants' abilities to smell? Which is true about newborn infants' ability to smell? A, they can recognize their mother's scent. And you probably know that answer from something we just discussed. B, they can recognize the scent of their amniotic fluid. C, both A and B. Or D, none of the above. Too easy, but you get a free question, so let's go ahead and launch it. Go ahead and answer. Best of your ability. Don't worry, this doesn't count towards nothing. Be proud to fail on your practice quiz. All right, I'll give you another 20 seconds or so. A few of you still haven't answered. Okay, that's great. Yeah, most of you got it right. The correct answer is in fact C, both A and B. So as we just discussed, and we discussed last class, infants can recognize their mother's scent. In fact, as you saw, an infant can kind of shows maybe something like a influence of a releaser pheromone and seemingly crawl towards a scent sample of their mother's. But we talked about that also in class last week. Yes, they can absolutely do that. And they can also recognize their amniotic fluid. They do show a sensitivity to that, with regardless of how it's actually tested, whether they'll suck on a pacifier faster or something like that. They do show some sensitivity to the scent of their own amniotic fluid. Okay, which might have some, might be related to how they're going to recognize their mom, which is contributing to the amniotic fluid. Her metabolism is going to contribute to the amniotic fluid as well. So both A and B are correct. Okay, so the correct answer here would be C. Again, there are, the none of the above Answers are often in the test, as well as all the above. Careful when you take the test not to be distracted by that. Don't assume that's always going to be the correct answer. Okay, so the correct answer is C there. Any questions on that or anything that we've been discussing so far? No? Cool. Okay, then I think we're ready to move on to more of this pheromone discussion. I think you're going to continue to find this stuff interesting. I'm going to share my screen here again. This is working. All right, hopefully we're okay. You can see me. I can't see me. That's all right. Yeah, I have no idea why I can't see me, but that's okay. Now, all right, I can't see me. Hopefully you can see me and everybody's happy. Let's continue to talk about this then. Where did I put my thing here? All right, so we're going to talk about now not singular pheromones, but the other type that Martha McClintock suggested, and that is modulator pheromones. All right, so let's just remind ourselves of this a little bit. What's going on here with the modulator pheromone is it's going to be something that induces a, sorry about this, folks. It's going to be something that induces an effect, but a very subtle effect. It's going to be kind of maybe the human version, or at least the subtle version of a releaser pheromone. It's going to induce effects, okay? It's going to induce maybe, mood effects or something like that, or, some much subtler effect than a, major type of behavioral reflex. But it's still going to be influenced by the chemical signals available in a sweat sample that can then be used by the smell system. Again, we're talking about all this stuff being unconscious. We do not know that any of this information comes, we're not conscious that this information comes through smell as we're receiving these signals, okay? But our, as we said, our smell brain is much more sophisticated and is much more consistent and continuous in its processing of this information than our consciousness would reveal to us. So what would consist of modulator effects? And we've already talked about one of these, and that is this eye blink response. So remember we talked about this fear-inducing smell. So you take sweat samples from somebody who's on a roller coaster or is about to take their medical boards or something, really anxiety-inducing. And you present it to naive subjects. And again, they're naive to the point where they don't even know they're getting a smell or a smell that's relevant to this. And what you find is that fear-related subtle behaviors seem to occur with these people. And we talked about the fact that they tend to interpret more neutral expressions as more fearful or angry or more negative. And then we talked about the eye blink response. And this is like a reflex, but let's make sure we understand what induces the eye blink response. What induces the eye blink response, oh, there's my toy over there, I won't bring it out. What induces the eye blink response is a sound, you blink, but the degree to which you blink can be influenced by an exposure to a fearful smell, okay? The reflex is induced by the sound. Loud sound that you're surprised by, somewhere near your face, okay, you are going to blink. There's the reflex. But the interesting thing with regard to smell is the magnitude of that reflex, how quickly it happens and how dramatically it happens, how, the amplitude, the intensity of that eye blink can be subtly affected by being exposed to a fearful smell. That might be a nice example of a modulator pheromone, right? So it's not inducing a reflexive behavior, but it is having a subtle effect on another behavior, that reflexive behavior, right? What else? What else might count as a modulator pheromone? Okay. Well, this is all new now. There is some evidence that exposure to a man's smell, okay, that is, related to their sweat, can, okay, elevate certain aspects of a woman's, I guess, mood and metabolism. All right, so this is a situation in which, what I mean by putative here, by the way, is just so it's not too confusing. For our arguments, we can consider it a woman being exposed to a man's smell, but they actually do some chemical kind of distillation of the man's smell that is possibly considered to be a pheromone. Okay, so that's being presented to women. They are unconscious of that smell being presented. It's kind of masked by a rubbing alcohol solution or something like that. They know nothing about being exposed to a man's smell. Still, it does seem to have very subtle effects, very subtle effects. Okay, it can change a woman's mood. It can make her mood a tiny bit more positive. It can increase her heart rate a tiny bit. Okay, it will induce a little bit more skin moisture. And I don't know if you're familiar with how that's measured. There's something called the galvanic skin response. I don't, it's not important, the details of this, but basically it's, kind of a measure of very subtle sweating on the skin. Okay, sweating that you wouldn't be able to see or necessarily even feel if you tried it on yourself, but it can be tested with a very sensitive, kind of device that's put on your finger. The skin moisture in a woman will increase a tiny bit if she's exposed to the smell of a man, and cortisol will increase a tiny, tiny bit. Okay, all those things have been shown to change based on a woman being exposed to a man's smell, or this distilled chemical in a man's smell. Okay, all without her knowledge, all unconscious, the whole thing's done unconsciously. What else? Another experiment, the women in the experiment are shown arousing videotapes, romantic, with, some of them bedroom scenes, that sort of thing that, women generally rate as arousing. They'll watch the arousing tapes and then report their level of arousal. Okay. If in fact they are presented undetectable smell of a man, their self-reported level of arousal will be a tiny bit higher. Okay, that doesn't mean they are being initially aroused by the smell. Ain't working like that. They need to be aroused first by watching these clips. And if they are in fact in the group where they are being exposed to the undetectable smell of a man, their level of self-reported arousal will be a little bit higher than a group of women that aren't exposed to that smell, but are still watching the videotapes. The videotapes are inducing the initial arousal, certainly, no question about that. But the level of that arousal, as self-reported by these women, can be subtly but systematically influenced by the presence of a male odor in the room. Okay? Now, let's step back, because I know you're thinking, oh my gosh, maybe I should be, if you're a guy, you're thinking, well, maybe I should be wearing a pheromone cologne or something like that to get women, romantically interested in me or something like that. It really isn't the way to think about it for various reasons, but, moral as well. But smell is not going to do that much, right? And in fact, you could probably increase, a woman's arousal watching tapes by doing other things. You could, accompany the videotapes, the arousing videotapes with romantic music, or turn down the lighting, right, of the room or something like that. those things would also, increase the woman's self-reported arousal, right? That doesn't mean that, that she's going to make choices she doesn't want to make or anything like that. Maybe, you walk in what we used to call bachelor pads. I don't know if you know this. Back in the 60s and 70s, men would have these bachelor pads that were, supposedly going to put women in the mood for a romance, right? And they'd have like water beds, or those were very popular back in the 70s, or circular beds that rotated. If you ever watched the Austin Powers movies, he's got one of those. Those are kind of supposed to set the mood for romance. Those work about as well as adding the smell of a man. So the way to think about this is not like a releaser pheromone by any means, but like like the same sort of, kind of heightened arousal that occurs with maybe lighting candles, maybe turning down the lights, or maybe putting on Marvin Gaye music. It's funny. I used to ask you folk your age range what type of romantic music you like listening to, and I guess you guys like Marvin Gaye too, which is great. So I'm still glad I called that chapter Marvin Gaye for your nose, because that's what it's like. When we were in college, we'd put on Marvin Gaye or something that was very, kind of, romantic and kind of smooth sounding when, we were in the mood for romance. And that's how to think about the added odor of a man when it comes to affecting a woman's behavior. It's not going to change behavior dramatically or anything. It just maybe increases the romantic mood a little bit. Okay, which is, I think, probably the better way to think about it. All right, what else? Well, there are other examples where a woman's, level of arousal or her degree of sexual fantasies will increase, and it's one that you might not really think about as romantic. If a woman is not exposed to the smell of a male, okay, but instead the smell of a nursing female, right? This is interesting. I mean, this seems kind of counterintuitive. And these are self-reported heterosexual women. Smelling the smell of a nursing female, I didn't put that in this slide. It's in the book. Read it, it's interesting. It does, smelling a nursing female also does the same type of thing, okay? Which is a little surprising. It seems that it can increase arousal, it can increase sexual fantasies a little bit, it can increase self-reported desire being exposed, not to a man's smell, but to the smell of another woman who happens to be nursing. And what might that do? Well, that might have maybe subtle benefits on, coordinating pregnancy and nursing between women, which could have evolutionary advantages. Certainly, we know that it does in animals. When mice are able to have offspring together, they can, share protection and nursing potentially if they have to, and all that sort of stuff. It might still be like a holdover in humans to have those sort of, kind of influences, again, so subtly that it doesn't mean that you're going to, have a baby right away or anything like that, but it does seem to increase arousal just a tiny bit. Cool. You can read more about that in the book if you want to. Okay. So let's now conclude about this. And there it is. There's some of that pheromone concentrate I said you can buy on the internet, but don't, because it really isn't going to do anything for you at all. When it comes to thinking about human pheromones, right, it seems that there is the best evidence for the type of pheromones we've been categorizing as signaler and modulator types. So, through the T-shirt test, it seems like, that the smells that come from one person can signal things like, whether we're related to them or not, what their gender is, whether they're a romantic partner or not, whether they have a genetic makeup that is complementary to our own, right? Those are examples of signaler pheromones, okay? Again, the type of behaviors those induce are nothing like a reflexive pheromone, nothing like a reflexive. okay? And also, it seems like there might be some evidence for these modulator ones as well. Again, another type of pheromone influence that's more subtle, right? Not going to smell a fearful smell and run away. You're going to smell a fearful smell, and it's going to have subtle modulator influences over a reflex that's already been established through sound, okay? Or maybe change mood a tiny, tiny bit, maybe increase cortisol levels a tiny, tiny bit, right? But nothing reflexive. So yes, we're talking about, things that can change in a woman based on smelling a man's smell, as an example. But again, nothing reflexive like that. There is this example of a primer pheromone, if Martha McClintock's research is correct, and then I think it's pretty compelling that she's able to shift menstrual cycles in these women who are getting the smell of other women presented to them in nicely systematic ways. So I think their menstrual entrainment is something real. Now, certainly it's probably not going to be determinate, right? There's other things that can play a role in menstrual cycles of women. It's not as, doesn't seem as systematic as what happens with mice or deer or something like that. But there is this influence, certainly. But when it comes to releaser pheromones, I think it's pretty clear that that sort of thing doesn't really happen with adults, at least, right? Maybe this infant example where they crawl to the smell of their mother, we can kind of consider it a releaser pheromone. Of course, we don't know the experience of the infant well enough to know whether we should consider that a reflex or consider it a choice or whatever. That's maybe not all that important. Certainly for adults, we don't get the sense that anybody is going to dramatically have a reflex that's like running away when we smell something scary, or smell another person being scared, or going into some sort of mating stance when we smell a male's odor nearby. We don't show any evidence for that, certainly as adults, right? But what about animals? I mean, are we really kind of distinct from animals in this way? I mean, do we know that these pheromone influences are in fact reflexive? Certainly they seem to be, right? But of course we don't know the experience of animals, the kind of thought processes of animals. When we see an animal behavior and we see it happen again and again, we call that a reflex. We call the ant scattering a reflex, and we assume because of their little brains that, that's what we got to call it. The same with a pig. Pigs seem like simple animals. If they go into that mating stance, that's a reflex, right? So what about animals? Well, what Martha McClintock and her colleagues point out is that this idea of a true releaser pheromone that induces these dramatic reflexive behaviors in animals all the time, consistently, whether that really exists. She says that she's not sure. She's not convinced that these are truly the sort of kind of automatic reflexive behaviors that they're portrayed to be in the biology literature, the biology research. So let's go to pigs, for example. That mating stance in the pig, okay, will happen if there's a male nearby. But as it turns out, the female needs more than the male's smell. So you look back at this research, and all that research had been done not with, a male sampled odor given to the female, but the male being present at the same time. In fact, the female needs more than the smell to show that reflexive mating stance behavior. They need something like the sight of the male pig or the sound of the male pig. And so they need, I guess what you could say is they need more evidence that there is a male pig nearby to go to the stance. Once they get that evidence, yeah, they'll show a reflexive behavior. So, the actual mating stance is, a dramatic change in behavior that seems to happen spontaneously. But the idea that it's necessarily only induced by the smell, that all you need is the smell to induce this behavior, might not be correct. Okay, you might need something more complex. And she has also a similar explanation of ants that I won't go into now. But what she's saying is, it could be that part of the reason we didn't have an idea that people can have these subtle influences or have, I guess, a sensitivity to pheromones might be because of what we assume are reflexive behaviors in animals that might not be as reflexive as we've assumed. Okay, we don't know what's going through their little pig heads. We don't know what their experience is, but it seems like it might be more complex than just, oop, they detect a smell, bam, there's the reflex. It could be that they need a little more information there, right? And she thinks that there's good evidence for that with the pigs. So it could be, certainly for going back to humans, we can say this, that the influences of these different types of pheromones are, they can be subliminal, they can signal things, they can modulate things, but they are not determinate. They are not determinate. I mean, the way to think about this when it comes to humans is to think about it as something like Marvin Gaye, right? That, yeah, it can kind of heighten the experience a little bit, right? If it comes to romance. What else? Like when we were talking about these smells that might, kind of invite people to spend more time at a hotel, or to like the lobby of a hotel more, or maybe gamble there a little bit longer. The way to think about the smell is not, some sort of kind of coercive influence. It's just like it's part of the decor. It's part of the decor, like, seeing the flashing lights and seeing the giant fish tank behind the front desk, or hearing the, nice music or something like that. All those things have a bearing on whether we stay in the casino longer, will we stay in the hotel lobby longer, whether we're in the, mood for romance. All these different sorts of influences. The thing about smell is that after a while, you stop detecting it. So you don't, maybe you're not aware that smell is also playing a role in wanting to stay, but it's the strength of the influence is nothing really more than the decor that's being provided by things you might be conscious of. Like, what a pretty fish tank, I'm going to hang out here longer. I like the music they're playing here. I like the Beatles, and they're playing a lot of Beatles music here. I'm going to hang out in the store a little bit longer. You also might like the smell initially, but then not be able to detect the smell, but your brain continues to like the smell, even though it's no longer conscious. So there's nothing really scary about smell. It just doesn't have, the conscious access that these other sorts of subtle, kind of contextual environmental influences do over the things that make us stay in a store longer, or maybe put us a little bit more in a romantic mood. I turn down the lights and make it more romantic. Those are all the same things as how smell would have an influence on us. Okay. Cool. Any questions on smell? That finishes our smell discussion. We can now do another test question practice before we take our break. But let's see, there are some chat questions. Let me take a look here. Okay. Do we see differences in groups of bisexual or gender fluid women? That's a really, really good question. And I haven't kept up with this literature over the last 5 years or so, but there did seem to be some emerging evidence that there are pheromone-like influences at the same kind of subtle level, at least on homosexual males. There was something about men wanting to sit in seats that had been occupied by other men that showed body symmetry or something like that, I think. Okay, but I haven't kept up on that research. I'm sorry, Ivy. I know that there was more research emerging about that population and many other populations, gender and identifying populations and things like that. Okay, any other questions? Okay. Then let's do one polling question, and then we'll take a break. Yeah, this one's easy. I'm going to skip to this one. All right. Yeah, this is a good one because people often get this wrong. And I actually was very careful because I knew this question was coming up. I was very careful in this discussion over the last half hour or so to make this clear. But let's see how you do, and then we'll discuss it. The startle reflex. That startle eye blink reflex is A, can be induced by a smell alone, B, can be induced by a sound alone, C, can be amplified by smell, or D, both B and C. Okay, the startle reflex, this eye blink reflex, right, we've been talking about, can be induced by smell alone. Can smell induce it on its own? Can sound induce it on its own? Can it be amplified by smell? Or both B and C true? Okay, go ahead and think about it carefully. You can look back on your notes if you want. You're doing good. Give you another 20 seconds or so. All right, I think we can stop it there. You did pretty well. And the correct answer is in fact D. And so a majority of you got this. So the startle reflex, and I said this before, this is very clear that you can't induce a startle reflex by smell. And all of you knew that, right? That's not the way smell works in this context, right? It's not all like you're going to blink, you're going to be exposed to a fearful smell from somebody, and your eye is going to blink. It doesn't work like that. Can it be induced by sound alone? It absolutely can be induced by sound alone. And in most laboratory experiments where the startle reflex is induced, it is induced by sound, that loud sound, whether it's a clap or wood being slammed together, or some sort of loud beep on headphones. That is most often how it is induced. However, because of what we've learned about the kind of modulator aspect of smell, it can also be amplified by smell. So smell can't induce the reflex itself, but it can amplify the reflex after sound, as an example, does induce it. So C is also correct, which means the final correct answer would be D. Both B and C are correct. And a majority of you got this. Okay, any questions on this quiz question? Okay, what time we got here? 11:28, so 10 minutes, so 11:38 or so, we'll come back and we will start our discussion of taste. All right, see you later. All right, we are back. Hope everyone had a nice break. Yeah, I don't know what's going on. Hopefully you're able to see my image while I'm lecturing. I am not, which is fine with me, but probably harder for you folks. Any question on anything to do with smell? I can also take a question here about the exam or anything like that before we continue to our next topic. Anything? No. All right. Then what we're going to start talking about is taste. And let me go ahead and share my screen. Boy, I sure hope the sound works on this. Okay. All right, hopefully you're able to hear this okay. Let's see your application. All right. So I didn't know very much about taste when I started writing the book. As I said, most of my experience and research is with multisensory perception and auditory perception. So we'll obviously be spending a lot of time talking about those things soon. But smell and taste, I did just a tiny bit of teaching during my regular perception courses, and went then and had to learn a whole bunch about taste to talk about the topic. And I think taste is really fascinating too. I was so glad I had a chance to do that. The way we're going to understand taste, though, is a little differently from how we understood smell and hearing, in that we are going to understand taste in its context with the other senses. Because in some ways, I think taste is our most, I guess, multisensory sense, in the sense that it is being influenced by all the other senses. You've already learned about how taste is influenced by smell, hopefully in your sections. But we're also going to find out that it's very, very influenced by touch and hearing, and, as we'll discuss right now, seeing. And I wasn't quite aware of how dramatic this is. Smell, I knew that smell influenced taste, and that smell was, responsible for a lot of our experience of flavor. But I didn't know about the other senses, and I think you're going to find this really interesting. One of the ways I kind of got exposed to this is I decided to go to a restaurant that exists in LA. I think it's now moved to Santa Monica, but it is what's called a dark restaurant. And this is a restaurant that on the inside is completely dark. You see nothing. In fact, they go out of their way to make sure that there's no light creeping in whatsoever. There's certainly no windows. And even where the door is, the door can open and let new customers in without letting any light in at all. There's a whole bunch of curtains and kind of passageways and things like that, so that once you're in, you've got no vision whatsoever. It is the darkest place I've ever been to in my life. I've been to, through caves and stuff like that. And oh my gosh, this was scary. It was like, your eyes were closed even while they were opened. It was that dark. And it took some getting used to. But what I was most interested in was whether it would have an influence on the food, because that's how they advertised this place. This place, well, you're going to see the little expose on this in the video in just a second. But they advertised it as a way to not be distracted from your food. Okay, so that was the idea. And by the way, this is kind of become a little bit of a fad. I think most major cities of the world now, Europe certainly, have a restaurant like this. And it's advertised as a way for you not to be distracted by other people in the restaurant and to really concentrate on the flavor of the food. And the question is, does it allow you to do that? Do flavors taste more vivid when you go and eat in a dark restaurant? Should we be eating at home with blindfolds on, when we're having a good meal or something like that? Does it allow us to taste the food more vividly? So let me show you a little video, and hopefully you can hear this. And if you can't, let me know by putting up a chat.
came here tonight because it sounded different. They said it's dining in the dark. It's probably a little unique and
Oh my gosh.
And disturbing, but that's why we're here. Welcome to Dining in the Dark. Y'all make this special.
Dining in the dark, it's a dining experience in a literally like dining room, dark dining room where all the food is served by blind servers, and that just makes you have like a totally deeper, greater understanding of the other senses that we all have, but often neglect.
Yeah, it's in the middle of the table, which is here.
The whole idea of dining in the dark comes from Germany. Ever since that first restaurant got so popular, so trendy, so like the thing to do, they started sprouting all over Europe. I took an American friend and she just said, like, why is the good stuff always happening in Europe? Why do we Americans always have to travel to Europe to actually do fun stuff? And then we just like started talking about it, and that's how we came up with the idea to do this in the United States.
OPEC dining, blind leading the blind. What the awesomeness of it is, although they can't see, I can guide them into a great evening of dining. Right. Someone is entrusting me, a person who cannot see, to guide them and lead them into a dining experience.
The server takes them to the table. The servers exactly know where they're going. So the diners, as soon as the door shuts, they're lost. The server really acts as a guide for the entire night. The blind server is like the only person that can actually do the job, because they have like had that visibility, which now becomes their ability.
I can't believe it. No, I know they're still late. I'm able to listen.
I'm not sure. Yeah.
People really go in there and just have such a blast. They just laugh. They just like they behave like kids.
Where are you?
Are you trying to feed me?
Yeah.
You're awesome.
Warm, cold.
That's awesome.
I love the way you look right now.
At first, when I was walking in, I'm like, I can't do this. I know I can't do this. But then once I got settled, I don't know, it was just crazy. You just like adapt pretty quickly.
There's complete suspension of self-consciousness. It just brings everything else into a sharper focus. I'm on your right, Brad.
When you go to a restaurant, what's the experience? And with obviously the ambiance, you know, to be seen in there. You don't experience that here. It's more really tasting the food and people's conversation.
This is like an experiment at being born. It's like it's genius. It's like it's a lot.
You're really tasting things, you're really tactile, you're really feeling things, you're really hearing things a little more acutely than you normally would.
Pretty tasty. It's good. It was incredibly hard to cut the steak, and then I noticed after a while, I was like, well, nobody can see me, so I just picked up the hunk of steak and started chewing it like a caveman.
Hey, Michael,
Yes.
would you mind helping us and escorting us out?
Yes, I will. It's almost indescribable unless you're actually, doing it.
Gives your eyes a definite break and allows you to focus on anything else. And you have your food, which is always a good thing.
Right.
Coming out was like very humbling for me. Like I feel very humble and kind of just appreciative of how easy life really is in a sense for me.
Hey, I'm glad I got skiing. All right. Well, come back again.
We will.
More than welcome.
Thank you so much.
Yeah, Michael's great.
Okay, that gives you a sense of what it's like. It was some of the things they said are absolutely true. You kind of do feel like a kid again, because it's such a unique kind of new novel experience. And you do have to kind of touch your food to know, where the food is and all that. And let's see, what else can I tell you? Well, some of the description in the book is, kind of gives you a sense of, what it's like for the folks that are eating there, and you can, hear from a little bit of the interviews. There's some folks don't feel very comfortable at all when they first go in. It is a little bit, shocking about how strange it is. And people do come out, say, I can't do this right now. I don't feel very comfortable. Can I leave? And then, when I was coming in, when me and my friends were coming in, there was somebody there with a glass of wine sitting in the lobby, just like almost shaking and saying it was too much for her. I guess most people are able to go back in, but at least one customer a night, they said, has to leave and can't eat there. It's just too uncomfortable. But the folks that feel comfortable, I have to say, it is a little weird at first, but by the time the evening was over, I felt so relaxed. There's just something about not having the visual distractions that really, kind of made you feel kind of more at ease. There wasn't kind of, being distracted by other people and other things that were going on in the restaurant. And by the end, it was very relaxing. So it was interesting in that way. I think I described this in the book, but what they do before you go in, they will give you the menu if you want it. And you have various ways of choosing. You can first say, I'll let the chef give me whatever I want, so I won't know what I'm eating until I actually have to taste it. Or you can completely order everything and detail off the menu what you want, all the different courses. Or what I did is I kind of just selected, like a category. I said, well, I'm going to feel most comfortable with the vegetarian sort of meal. So that's what I ordered. I remember eating it. We got bread and beer and hors d'oeuvres, our first salads and whatever. And then when my main meal came, I remember really not knowing what was in it. I mean, it was clearly, there was a coconut kind of taste. There was rice, but there were, like objects that were on the plate that I didn't know what they were. And I remember a number of times picking, like putting my fork into things and then, bringing the food up to my mouth and missing my mouth and getting speared by, I guess it was probably a green bean at one point in my cheek. And then at one point I felt something a little thicker. I put it in my mouth and I had no idea what it was. It was long and thick and about the size and shape of like a finger. And I thought to myself, oh my gosh, maybe the chef is blind too, not true, but it turned out to be a carrot, I think, which I figured out by the end. But you really don't know what's in your mouth until you're really kind of chewing it and you're experiencing it. So, the question is, what's this like in terms of the flavor of the food? That's really what I was after here with this experience. And what I'm going to do is I'm going to, before I give you the answer for that, I'm going to, let's pose the question, does food taste better in a dark restaurant? I'm going to look at the research and then see what we can predict if food tastes better in a dark restaurant. However, before I go into the research, are there any questions about this restaurant? This restaurant, I should say, does exist. It has moved. When I went, it was on Sunset in Hollywood. Right, I think the Hyatt, it was in the Hyatt or the Hilton Hotel, but it might have said in that video, I don't remember. But it has moved now. It's in Santa Monica somewhere, and I think it's still called Opaque if you're interested. It is expensive, but it might be something to experience at some point with some friends. It's a real experience to go in. Are there any questions about the restaurant? You can either ask out loud or through chat. I'd be happy to answer questions you might have on this. No? Okay. All right. If anything arises, feel free to interrupt at any time. But if not, let's go to the research. And I mean, because this is what happened to me. I mean, I after I ate there, I came back and I said, okay, now it's time to do look at the research on flavor and how it's influenced by sight to see what it suggests. And it's pretty dramatic how seeing your food influences how it tastes. Okay, so a lot of this research, by the way, when it comes to sight and flavor, is done with fruit drinks, because fruit drinks really can be created. And we're talking about, I don't know, like power drinks. Actually, that's probably your best reference here, like the Powerade and different types of Gatorade. The flavor really, I mean, there's a lot of flexibility in how the food scientists create this. They don't have to necessarily use the color that's associated with the flavor, right? They can mix this up. And this is research done before, I think, Powerades were popular, but it was done with like sugary drinks. I don't know if you guys had Kool-Aid or anything like that. It's done with that type of drink. But what they found very, very quickly is that they can change the experience flavor of a drink like this by changing the color. So they can, mix together what should taste like an orange flavored drink and put in a red food coloring to it, and then ask people what flavor they're drinking. So they hand them the glass, it's all red, and say, okay, could you taste this and tell us what flavor you think it is? And they will taste it, and they will likely say that it is, sorry, before I put that up, they will likely say it's cherry, even though it's the, flavoring compounds that are there for orange. And the opposite works as well. You can take the cherry flavored drink and put food coloring in that changes it to orange, and people will sip it and say, oh yeah, that's your orange drink. Even though all the chemicals in there that should be affecting their tongue and their smell system should be for cherry. So these examples show that what color the drink is has a strong influence on what you believe the flavors are. So fruit drinks taste depend on color, and this is something they clearly know as they're making the Powerades. And it's very relevant to this fact that the intensity, not just the color, red versus yellow versus orange, not only does that determine the flavor, but the intensity of the color, how bright the color seems to you, what we call the color intensity, will determine how strong the flavor is, right? And this is something they absolutely test when they're doing, creating Powerades and trying to determine how it should look, as well as how the flavor should, how the chemical compound should affect your tongue and your smell system. Is that they will, if they, they will give, maybe what's your favorite? My kids like the dragon fruit flavor. They ask me to get that for them all the time. If the dragon fruit flavor was a very dull color, it's actually a very bright color. If it was a very dull color, it, based on the research, would taste to them less flavorful. It would seem more dull, not very sweet, not very much like the dragon fruit flavor. And then all you have to do to make it seem more like a dragon fruit flavor, or whatever that is, or a stronger flavor, or even sweeter tasting, you just change the color. And then people will say, oh yeah, that's good. That's a really nice, strong, flavorful drink, even though nothing that's affecting your tongue or your smell system, which also is going to affect flavor, nothing has changed in the chemical compound, only the color. In this case, it's the intensity of how bright, what is it, red or whatever, how bright or red it is, makes a big difference. And this is not just true of fruit drinks. It's been tested. Very often, these sort of experiments are tested with things that are sweet, that people can, that don't mind, having in a laboratory or whatever, like custards and cookies and cakes, things like that. The color, the color brightness, all have a bearing on how sweet these things taste, how flavorful they taste. And again, nothing's changing in what's touching the tongue or going into the smell system. The only thing that's changed is the visible appearance. And there's some kind of sneaky ways of doing this where, with a custard, what you can do is you can have like this kind of secret bowl that actually, oops, there we go. A secret bowl that actually has two compartments. There's going to be the custard that you're seeing on top, And then there's a secret compartment here where there's actually another custard on the bottom underneath what you're seeing, and they're separated. But what you can do is what they'll do is they'll put a straw into it. So what's going into your mouth is the bottom custard, but what you're seeing is a different custard from the one that's actually going to go through the straw into your mouth. So it's kind of got these kind of secret chambers in here. And what they can do is they can change the appearance of the upper custard. They can put a thicker custard or a thinner custard, or one that's more yellow or beige or whatever it is, without changing the bottom. And that's a way they can test how the appearance of the custard independently influences your flavor. And what you find is that that the visible texture, so whether it's a thick custard or a thin custard that you're seeing, influences how you're able to taste the custard. And it says here, facilitates discrimination of custards, whether you're tasting two different custards or a single custard twice. If the texture is different, it makes it a much easier sort of task. So using these kind of trick bowls, they can show very easily that what you're seeing will influence your experience of the flavor, including the sweetness, including how appealing it is, all that sort of stuff. And so, oh boy, does what you see influence what you taste so strongly, which is going to give us some hints about what happened to me in the dark restaurant. Okay. Before we get to that answer, though, let's do a little bit more of the science and talk about some of the neurophysiology. Oh, by the way, I should just mention, all these effects are truly perceptual. What I mean by that is that we have ways in the laboratory, in the perceptual laboratory, of determining whether people are kind of giving us their kind of opinion about something that's maybe more rational, maybe more I guess intellectual, versus a truly perceptual effect. So in this case, for example, let's go with the custard case. It could be that the subjects are kind of second guessing the experiment intellectually and saying, well, I bet this is on appearance, this experiment, and so I'm going to kind of give the experimenters what I think they want and show and pretend like the flavor is changing with the color or something like that. So we have to make sure in the laboratory that that's not happening. We want to make sure that people are really having a fundamental perceptual experience without all this thinking that's being influenced by what they see. And there's ways of doing that. And clearly, what's very clear in this research is these are truly perceptual effects, not like afterthoughts of the subjects in the experimenters, something like that. Because sometimes when you talk about this research, maybe people are in these experiments are being a little bit more introspective and trying to figure out what's going on. And that's the sort of results you're getting. These are real perceptual effects. Okay? And we believe that's because of the neurophysiology is so clear on this sort of thing. So there's a ton of cells in the both the visual areas and the tasting areas of the brain that overlap and show that they can be induced both by seeing food and tasting food. So there are lots of cells dedicated to kind of the joint influences of what a food looks like and the food experience on your tongue. It seems like the brain's kind of set up to do this kind of integration of these two senses. And even when you smell food, you don't actually put it on your tongue, but when you smell food, you can induce responses in the visual areas of the brain that are pretty systematic. So again, now these are cells in the area back here that's typically responsible for detecting light. But in these cases, you're not seeing anything. Your eyes are closed, maybe blindfolded. You're smelling food, and it's as if your visual brain is getting stimulation, which is really interesting. And by the way, just as a little aside here, I think that maybe this kind of influence of vision on taste and maybe smell on vision, all that sort of thing, is one of the reasons that these kind of cooking shows and cooking competitions have had so much success. Because if you think about it, kind of objectively, why should watching people cook and watching what they make be an appealing thing if you're not tasting the food, if you're not even smelling the food is nowhere near you, but you're seeing the food. And I think that we have such a, kind of strong connection between what food looks like and how it tastes, that we're kind of tasting it vicariously in some ways, because our brain is kind of set up to do that, right? Your mouth waters when you see food, right? That, probably some of you folks, your generation's much better at cooking than my generation was. It's great. Something that how the food looks on the plate is going to have some bearing on how appealing it is. So now let's get back to the dark restaurant. What happens when all that visual information that our brain seems set up to use to influence our experience of flavor is taken away? What happens when you eat with no vision? Okay, at least like in this context. And I'll tell you what happens. Okay, I'll tell you my experience. And the experience of my friends. I was there with my wife and another couple. We got, I think the first thing they asked us was our drink orders. We all got some drink. I got a beer that I've had many times before, Sam Adams or something. I don't remember what it was. They didn't have great beer selection. Anyway, I remember tasting it and saying, that's weird. It tastes a little bit bland, the beer. This is a beer I've had before, and it tastes bland. Maybe it's not cold enough, or it's not the right temperature. Okay, fine. But my friends said the same thing. They got kind of a little bit of a fancier drink, maybe apple martini or something. I think my wife got or something, and she said, yeah, I think this is just way too weak. Okay, first thing. Next thing, they bring out the bread, and the bread's fresh baked, and it's warm, and that feels good. Warm bread in your mouth when you can't see anything is very kind of, it's kind of a nice feeling. But after a little while, it got a little cooler, and we said, this bread doesn't have a lot of flavor either. Okay. And then after that, we all got separate dishes. We all got separate, salads or soups, and we all got different entrees, and we all got different desserts, and we all had the same experience. We felt like the food didn't have a lot of flavor, which was a bummer, because at that point we thought, this is really kind of odd, because this is a pretty high-end restaurant. The chef has a good reputation. They took him from another restaurant, regular restaurant, but the food doesn't have a lot of flavor. It's kind of a bummer, because it's a cool situation. You're in the dark, and it's kind of a neat, novel thing to experience. It's too bad the food isn't a little bit better. But now you know why it wasn't better. And I think the biggest clue comes from this, that the intensity of taste depends on what you're seeing. And if you're not seeing anything, especially in a spontaneous situation like this, where you're going from being a visual person to functionally being a no vision person, food becomes more bland. And that's exactly what we found. Does food taste better in a dark restaurant? No, it doesn't. It tastes more bland. Two other things I wanted to say about this. If in fact, at least last time I looked at the Yelp reviews, it is something people comment on, that this idea that you're going to be able to taste your food more vividly because you're not distracted is not true. Okay. You're not distracted in the same way visually, but you're not getting that visual access to the food, which turns out to be important in having, a vivid, a nice, rich experience of flavor. What else? Well, we're going to be talking about some experiments. I think maybe even we might get to it in Tuesday's class, next Tuesday, where normally sighted people are put into a situation where they're going to be blindfolded for a couple of days. And one of the first things people comment on when they are blindfolded for a couple of days, and usually they're either put in a laboratory to sleep, so they can be monitored in a safe way, or sometimes they're put into hospital rooms if their brains are going to be imaged, just to keep them safe. One of the first things they'll say about being blindfolded is that food tastes bland. Okay, so it's known. I mean, this is absolutely known. So you're thinking, okay, what happens for a blind person, right? The blind servers. Well, if it's a person who's gone blind because of a traumatic brain injury or something like that, they will say that food tastes bland too. But after a while, they will learn to compensate with the other senses. So a blind person does not taste food in a more bland way, we think, because they learn to maybe include the sounds of food and the texture of food that they can feel with their tongue, and certainly the smell that's kind of going through the back of the mouth that you've learned about. They're learning to use those other senses, or if they're born blind, they'll use those other senses to compensate and not have the deprivation of flavor that occurs from not being able to see food. However, if you're blindfolded, if you're a sighted person, you're used to being able to see your food, and all of a sudden it's taken away, your experience will be of more bland food. So the answer is no. Food does not taste better in a dark restaurant. It tastes more bland. I haven't gone in a long time. I haven't gone in about 10 years, and it's a little bit of an expensive restaurant, so I haven't run to go back there. I'd rather go to an expensive restaurant where you can see your food. But I might go back at some point to see if they've done anything to kind of compensate for the flavor that's lost. Maybe they'll use more spices or something like that to kind of exaggerate those aspects of the food. But yeah, the idea that you can better taste your food in the dark, at least if you're a sighted person, it's not true. Okay, I see there's a question here. So let's see if we can answer. Is color relevant? Oh, it's a great question, Ivy. Is color relevance an evolutionary adaptation? Yeah, it probably is. Sweet things tend to have brighter colors. So maybe that for energy, for quick energy expenditure, all that sort of stuff. Yes, color relevance probably is an evolutionary adaptation in that sense. That we are going to be bright things are going to attract our eye and attract us using the food. Of course, there are situations where it can be dangerous. There are some bright color berries that are poisonous to us. But yes, I think there is an evolutionary component, an adaptive component to that. Absolutely. Good question. Okay. Other questions about the dark restaurant or about how vision affects food and flavor? Any questions about that? No? Okay. Let's go on then and talk about leave vision. Hopefully I convinced you that vision's important. You might be surprised to learn about this though. Not only is vision important, but sound is important. And some of this is going to be intuitive to you, but I think you're going to be surprised at how powerful sound can actually influence things. So, you're eating fresh vegetables. And the fresher the vegetable, the fresher the carrot, the fresher the celery, the louder sound it will make. And maybe then you wouldn't be surprised that our sense of crispness can come from sound. And so, yeah, depending on how sound is manipulated while we're eating, we can have an experience of freshness or crispness. And our sense of the freshness of a vegetable, for example, will be the same whether we're hearing ourselves crunch the vegetable, or if it's the person next to you crunching, or you're listening to somebody crunch a carrot over a recording, through headphones or something like that. Your judgment of how crisp, how fresh the thing is will be the same whether you're chewing it or whether somebody else is chewing it. Now, I need to tell you a little bit about what happens when you chew food and how it affects sound, right? So when you're chewing something, hopefully your mouth's closed during much of it, but maybe when you first, when you bite into a carrot, your mouth has to be open because a little bit, when you bite into a celery, if it's sticking out of our mouth, it's going to mean that sound is going to get to our ears in two ways. One of the ways is the most obvious to what we understand with sound is that it goes through the air around to our ears, and that's called air conduction. So, if I'm biting into a carrot like that, the sound leaves my mouth's open a little bit, and it, goes around here and comes to my ears, and that's how I'm hearing crispness. However, we don't, when something's happening with our body, we don't just get sound through the air. We also get sound through the vibrations that are conveyed through our body, our skull. Really, in this case, we're talking about our skull. That's what this is showing here, that sound, mechanical disturbances that are occurring in the mouth are conveyed through the bones that are connected to our skull that then get into the sensory organs of the ear. That is called bone conduction, okay? Now, we've all experienced the effects of bone conduction. Maybe you remember this, you were a little kid where, you know, you're used to hearing your voice and then you hear it on your own. You're used to hearing your voice and then you hear it on a recording, right? You hear your voice, you know, maybe on a video or an audio recording or something like that. And, you know, maybe on a message that's left, you know, on the phone or something like that. And it sounds thinner. It sounds a little tinnier and whinier than what you're used to hearing. And sad to say, that is in fact how your voice sounds to other people. So yeah, I have to listen to these recordings to make sure that they come out okay. So I'll always listen to a little bit of it. And my voice to me still at this age sounds a little whinier, sounds a little thinner than I experienced my voice on my own. And that's because I am getting my voice in two ways, which is different from how everyone else is getting my voice, which is just through the air. I'm also hearing my voice through bone conduction. You hear your own voice through bone conduction. And your own voice through the bone sounds more resonant. It sounds a little deeper almost. It sounds more, I don't know, authoritative or something like that. So when you hear how your voice sounds to everybody else, which is just through the air, and that's how the recording picks it up, it sounds a little thinner. So that experience is from not hearing your voice through bone conduction, which is kind of, you know, an illusion a little bit. You know, you're hearing your voice as being deeper, more resonant than it actually sounds to other people. Right. Anyway, while you're chewing, because you're, you know, potentially opening your mouth and closing your mouth, that experience can kind of flip back and forth between mostly sound of our chewing through bone conduction and then sometimes sound of our chewing through air. So it's going, you know, obviously when we first put food in our mouth, we're going to be hearing it through the air and through our bone. When we then close our mouth as we're being polite and, you know, finishing our food with our mouth closed, you're hearing it mostly through bone conduction. So it's going to change. So the relative importance changes as you chew. Okay. But here's an experiment that shows that by presenting a manipulated sound to people as they chew, it can affect how something tastes, okay? How fresh something is, maybe most importantly. And this is an experiment. I always thought this was very cool. This was done with potato chips, and then ostensibly it was an experiment where people were asked to rate how fresh or how stale the potato chip was. And I think they actually were also asked how much they liked it. And they went into a little booth and were given, they were blindfolded. Actually, this is, but I'll tell you about Pringles in a second. Here's the video. I'm sorry, not the video, the image of what's going on here. Somebody's in a sound booth. Actually, what they don't show here is people are actually blindfolded too. So this is all coming through sound. They are listening to themselves crunch through the headphones. And you can see there's a little microphone there. What that microphone does is pick up the sound of the chewing, takes it out to a computer, which then manipulates the sound and brings it back to the headphones. So people are listening to their own chewing in real time, but it's been a manipulated sound, okay? And people are told that they are given, they're being given different types of potato chips and they're different shapes. Now, the reason they use Pringles is because, you know, Pringles are always the same, no matter, you know, which can you open at any one time. You know, I mean, Pringles are, you know, they're the same shape and the same size and the same salt. I mean, you know, Pringles are the most, you know, I guess, predictable potato chip in the world. All right. Now they want people to think these are different potato chips. So what they'll do is they'll cut them into different pieces and hand them different size pieces and stuff like that. But, you know, as far as the tongue is concerned, as far as the smell system is concerned, they are getting the same potato chip again and again. What's changing is the sound, because the computer that is getting the sound fed from the microphone can be adjusted so the sound seems brighter in some cases. It seems louder in some cases. It seems duller and maybe more muffled in other cases as they're eating the potato chip. Okay, so imagine you're in the experiment, you have headphones on, you have a blindfold on, you're given a potato chip, and you said, okay, just give us a rating of how fresh this one tastes and whether you like it or not. You start crunching, and in your headphones, you're hearing yourself crunch. Cool. And it sounds really bright. It sounds very kind of loud and clear. And you would say, yeah, that is a crisp potato chip. I like this one. This one's, not stale at all. And then on your next potato chip, you're given a new sample. It's cut differently, so you're not aware it's just a Pringle again. You put it in your mouth and you start crunching. This time, though, what you hear through the headphones is you're crunching, but it sounds just a tiny bit duller. Imagine, kind of turning down the treble control of your car stereo a little bit. It sounds, you know what I mean by duller, like kind of a little muffled like that. You say, I don't like this one. This one kind of tastes like it's a little stale. It tastes, less flavorful even, and I don't like it as much. This one is not as appealing. And in fact, that is a clear influence of sound on taste. Okay. So yeah, it makes a difference. And again, it is, I guess maybe this is a little bit intuitive with regard to how crisp something is, but people, would also judge how much they liked it, and that was influenced as well. So if it's lighter and brighter, it will taste crisper to people, even though the only thing that's changing is the sound. If it's quieter, if it's duller, if it's more muffled like that, it will taste stale. Okay, that's how people report. And this is also a perceptual effect. It's not like, some sort of afterthought on the part of the subject trying to please the experiment or anything. This is, we have ways of determining if it's truly perceptual, and it is. People's experience of flavor when it comes to crisp, stale, appealing, less appealing, will be directly influenced by the sound they hear as they're crunching. Okay, so sound is also going to be one of these senses that influences taste dramatically. What else? Touch. Okay, and there's some really interesting demonstrations of this that I think you're going to find very compelling. And one just involves having a hard candy. Having a candy you can suck, or maybe a mint. Maybe the thing that you're maybe more used to is having a mint that you would suck on, or maybe a cough drop that you would suck on, or something like that. And say it's a candy now, for the hard candy, some sort of sweet thing that you would keep in your Lifesaver. You guys ever have Lifesavers? Maybe not. It's a hard candy that kind of floats around your mouth and kind of slowly disappears. It gets smaller and smaller, right? There's a hard candy. And your experience of it is that hard candy is going to have flavor, and say it's I don't know, let's make it a grape hard candy. Okay, it's going to have a flavor that's going to be the same flavor regardless of where it is on your mouth, right? So whether it's in the front of your tongue, whether it's in the back of your tongue, whether it's in the side of your tongue, it's always going to have the flavor of grape, and it's always going to be a sweet grape. Okay. However, it's interesting the way that your tongue works, and maybe you've learned this somewhere along the way. You don't have sweet taste buds spread evenly throughout your tongue. Most of your sweet taste buds are back here. In fact, there's none, I think, way back here. There's no sweet taste buds at all. And maybe you've learned this tongue map in other places. We have a lot of salty taste buds right here that are sensitive to the salt flavor. Sours back here, bitters here, and there's what is it, umami? Is that another possible flavor? I can't remember where those taste buds are. But we have different regions of our tongue that are sensitive to different, I guess, dimensions of these flavors. Now, clearly, the grape has to come through smell to some degree, right? Because this is only giving us sweet, salty, bitter, and sour, right? We know that smell is going to be an important part for this, and you can do this jelly bean demonstration where you close your eyes, choose a jelly bean in one of those Jelly Belly This is how we used to do it when we could go to the lab. We gave people jelly beans and have them put in their mouth with their nose closed, their eyes closed, and start chewing it, and they could taste sweet, maybe licorice, maybe if it's in there or something like that. But if it's just sweet, like a fruit flavor or something like that, they cannot determine what the flavor of the jelly bean is. They can only do that when they release their nose, and they're then getting the air from the back of their throat through their nose, and then they can taste grape. But the sweetness, okay, is only detectable by these taste buds up at the front of the tongue. However, the candy seems to have its sweetness follow it. Okay, so whether it's back here, it seems like, yeah, I'm tasting the sweet candy back here. Whether it's over here, I'm tasting the candy back here, even though your sweet taste buds are only there. So the flavor follows where the candy is touching your tongue, even though there's no sweet taste buds back there. And this is something that you can do kind of very methodically, right? So you can, rather than doing it with a hard candy, you can, have a subject close their eyes and stick out their tongue, and then take a Q-tip and put it in a sweet solution, and first put it on the front of the tongue so it can be identified as sweet, and then, carefully place that Q-tip in various locations of the tongue, including the majority of the locations that don't have sweet taste buds. There's none back here. Say, do you taste the sweet back there? And they'll say, yep. Do you taste the sweet back here? Say, yep, absolutely, absolutely. But there's no sense organs to taste sweet back there. What happens is where the tongue is touched will make the sweetness seem to follow the object. And this is sometimes called the sweet tongue illusion, if you're looking for a name for the illusion. But it's what's called also tactile capture. And where the tongue is touched will determine where the flavor seems to come from. Okay. Even though there's no sweet taste buds back here, it seems that you're tasting sweet back here because a candy, which has been identified by the tongue as sweet before, is touching back there. So our experience of the sweetness follows where the tongue is being touched. Okay, pretty cool. Let's see. There's another way of doing this. It's very, very, very quickly. There's an example in the book with a Q-tip where you kind of you rub the tongue along its edge after it's been identified as sweet, and you have the illusion again of it's staying sweet once it's been identified as sweet. Yeah, it works like this. I'll just do it very quickly. Okay, your sweet taste buds are pretty much only there in the front of the tongue and the very tip. Sweet right here. So what you do is you take this sugar solution Q-tip, start on the edge. Okay, start over here, for example. Oops. Yeah, start over here and rub it there. And people will say, do you taste anything? You say, nope, I don't taste anything. I feel you touching me there, but nothing to taste there. And then it gets to the front of the tongue. You say, oh yeah, it's sweet. And then you continue to the other side of the tongue and it continues to taste sweet. You say, okay, then all my sweet taste buds must be in the front and on the left side of my tongue. Well, guess what? You can do it starting on the left side and the same thing happens. Now, you start from the beginning, you have a new subject come in, you start on the left side of their tongue and you bring it around like this. You say, taste anything? Nope, nope, nope. And then as soon as the front of the tongue gets it and it's identified as sweet, and you continue to the other side of the tongue and say, yep, it's sweet all the way around to the right side of the tongue. So your taste buds for sweet must be on the right side. No, that's not it. It's an illusion. Once it's identified as sweet, the flavor seems to follow where you're being touched, even if there's no sweet taste buds there. Okay, that's what we're talking about. That's the illusion. Okay, I have this term blind spot analogy, and I'm actually not going to talk about it. I decided it's a little bit of a complicated situation, but it has to do with the blind spot on the eye. If you're interested in that type of thing, go ahead and read the book. I won't ask you a question about it. Okay. Good. So that's how important touch is. Touch can actually influence where flavor seems like it's coming from. All right. But there's other aspects too. So remember we talked about visual texture influencing what you taste. It can also occur with texture that's determined by the feel of the food in the mouth. Okay. So rather than, you can use custards again, rather than looking at the custards now that have different visible textures, you can have a blindfold on and have the custard just put right in your mouth, with a straw or something like that. And you're able to determine how thick the custard is, how thin the custard is, using your tongue, right? Your tongue has, it's sensitive to how things feel as well as how things taste. And the same is true of the inside of your mouth. And depending on how thick or thin that texture feels, you will then experience it as sweeter or duller, correct? Stronger sweet taste or weaker taste. And generally, what it turns out to be is that the thicker the custard is, the more viscous the custard is, the weaker the taste will seem, even though it has the same amount of sugar, the same amount of vanilla flavor, the same amount of whatever else egg flavor that goes into the flavors of a custard. Everything else will be absolutely the same, except the thickness. Okay. And food creators, food designers know this type of thing, that if they want to make a custard is thick, right? Because they think that that's just going to be like their home style custard, right? It's going to be thicker than a prepackaged custard or something like that. If they call it home style, chances are it's going to be a little thicker, maybe a little lumpier, as if it were made at home. When they do that, they know they need to add more sugar in order for you to have the same experience of flavor. Because once something is thick in your mouth, your tongue needs to kind of have more sugar to offset the experience of touching something thicker. Okay, so texture, felt texture in this case, not seen texture, we've already been through that, but felt texture in this case influences how strong something is considered flavorful, how strong something tastes. What else? Temperature. And this is, I'm sure, very, very intuitive to you, right? So when foods get sent back at restaurants, it's almost always from temperature, right? That's the only time, unless it's a terrible tasting food or it's really, really bland. I'll never ask, I'll never have the waiter come over, and I get embarrassed doing it anyway. And so I try not to do it. But if a hot meal comes out cold, I will say, would you mind heating this up a little bit? And that's certainly the most requested food change of waiters, is when the food comes out cold, because it can be a little unpleasant. But not only that, it has an influence on how flavorful full of food is. And so, good restaurants, careful restaurants, they will, time things well to get so that the food gets to the customer with the right temperature. And the right temperature is, pretty close to 98.6, actually. I think it's the same temperature as our tongues, as our tongue measurements. So that is going to be where they're targeting the food temperature, because our tongues are most sensitive at that temperature, most sensitive to food at that temperature. And even very small changes. So if a food comes out and it's not, say, 98.6, there's flexibility to some degree, but if it's like below 95, it's going to change the flavor. It also, if it's above, say, 101, 102, it's going to change the flavor. So, it better be over warm than over cool, because you can always let the food get a little bit of get a little cooler. I'm just thinking the same thing's absolutely true with beer. You don't want a microbrew, nicely made beer to be too cold. It doesn't have the flavor it really needs. You want it to be a little bit closer to your tongue temperature. Even if you don't like warm beer, you probably know that there's more flavor to a beer that's a little bit warmer than right out of the refrigerator. It's the same exact thing. But in restaurants, you want it to be certainly above 95 degrees and below 102 or something like that. Because just 2 degrees Fahrenheit in either direction is known to influence the taste. And it's not, it can influence how strongly something tastes, but it also can, change it and make it seem a little too salty or a little too sweet or something like that. I've had experiences where, beer. I'm going with beer because I actually I have a hobby of making my own beer, so I'm kind of become very aware of what goes into beer. That a beer can taste too hoppy if it's presented too cold, and it will end up being a more balanced beer in reality. It just has to warm up a little bit. So it's not just how vivid it tastes at the right temperature. It's also, what is the balance between the flavors, and how that's affected by temperature as well. And temperature can have such an effect on taste. You can actually have illusions. You can give people just water basically and say, okay, this water is mixed with a very, very, very light solution of salt or sweetness. You have to determine which one it is. And you give people a sip of water and it actually has neither salt or sweet in it. But what you do manipulate is the temperature. And this is kind of a phantom flavor. What I mean by phantom here is there's no flavor in the water whatsoever. You're kind of inducing it by suggestion a little bit. But beyond that, what happens is that there's some predictable outcomes depending on the temperature of the water. So colder water will in fact be more likely to be said to be salty. Okay, warmer water will be more likely to be said to be sweet. Now, again, it's just water. You're kind of tricking your subjects in this case. You say, okay, we added a tiny bit of salt or a tiny bit of sugar. Tell us which is which. Here's some water. You give it to them, it's cold. They're more likely to say it's salt. Instead, now you give it to them and it's warm, maybe a little bit warmer than the tongue. It's more likely to be identified as sweet. So you kind of do have, they're light, they're very light, phantom flavors, but it is predictable. Warm will give you a sweeter flavor. Colder will give you a salty flavor. And this is not surprising when you look at some of the neurophysiology. It is the case that the part of our brains that are sensitive to the parts of our brains that are sensitive to flavor are filled with cells that are sensitive to touch as well. So, again, our tongues are doing more than detecting the chemical compounds of whether salt, sweet, bitter, sour. Our noses are, of course, getting the rest of the flavor, but tongues and the interior mouths are also touch organs. And what they touch in terms of viscosity, in terms of location, in terms of temperature, have direct influences on flavor. So let's finish this conversation about this aspect of flavor and taste by talking about the big picture here. I think that taste is the most multisensory of our senses. I think without influences of touch, hearing, sight, and especially smell, okay, our experience of flavor would be very, very different. Okay, our experience of flavor would be very, very simple, very diluted. Okay, we would have salt versus sugar versus sweet versus bitter versus sour versus whatever umami, whatever that other flavor is, potentially, and nothing else. In order to have flavor, we need our other senses. Okay, and in that sense, this is true. All the senses of these have direct influences on taste, right? Even hearing, which is, I think, maybe the least intuitive of how this might occur. What we hear can influence how flavorful something is, as well as how crisp or stale it is, but also how much we enjoy it. They all have, and hopefully you got a very strong sense of that from everything we've discussed so far. However, one last thing to mention here is that the senses not only directly influence our experience of flavor, they can influence each other. And then that, kind of secondary sense can have an influence over what we taste, right? So they can indirectly influence taste by influencing each other. Let me just give you a very simple example of this. It is known that our sense of smell can have an influence over the perceived viscosity, right? So now before you taste something, you are asked to smell a custard, and that smell can then influence how viscous the food looks or feels in our mouth. And now it's just a smell that's influencing this. So stay with sight. The smell of custard can influence how viscous we say the custard looks. And then that appearance of the custard can influence how it affects our sense of flavor. So then smell is affecting sight, and sight is affecting our experience of flavor. That's kind of an indirect influence of taste, right? We know that texture, what we feel in our mouth, can affect perceived temperature, right? So how the food rolls around in our mouth can affect how warm or cold, I didn't say this before, how warm or cold the food is rated by our subjects. And then that experience of the temperature, of the perceived temperature, can also influence how the food tastes. So there's an indirect influence of the senses on taste as well. So it's like, I think I say this in the book, it's like your taste brain is like a kid with a chemistry set and is experiencing all these different influences to kind of get to the ultimate experience of flavor. And I think this multi-sensory aspect of it is why the cooking shows are so popular. You can get a lot from seeing the food and then having the people kind of describe the food for you. Your taste brain is ready to take in all this information and kind of have a sense of what something is going to taste like, which is really interesting. So I think taste is especially like this. Okay, we're going to continue with taste, but other aspects of taste in just a second. But let's go ahead and do a practice question. Oh, and there's some questions. Good. Chat questions. All right. IVS, I guess that's why people have water temperature preferences. Oh, when they drink water, it could very well be, right? So I like my water really, really cold typically. And I know not everybody does. I think it's also, partly experience. Let's see, do certain genres of music played in restaurants? Yes, it does. That's absolutely true. Certain genres of music played in restaurants affect how people enjoy their food. Absolutely. We will discuss that actually in just a second. And I think it's still in my lecture, and if it's not, it's in the book, but yes, the answer is yes. And we'll give the details in just a little bit. Great questions, Ivy. Thank you. Okay, so let's do a polling question. And I think we have a couple more smell questions here that are good. Yeah, this one's good. Yes, so this one has to do with men preferring a woman's smell, possibly depending on when in the menstrual cycle the woman is providing that smell. So the question is this. Men prefer the smell of a woman when she is A, fertile or ovulating, B, the day right before menstruation, C, during menstruation, or D, none of the above. So this is right from the lecture we gave about an hour ago. You can look back in your notes if you want. It's very straightforward. The answer is right there. And I'll give you a minute. I think it should be pretty easy for you. All right, another 10 seconds. Looks like a lot of you got this. This one's easy. Yeah. Okay. Sounds good, folks. You did perfect. The correct answer is A. That is the correct answer. When a woman provides an odor sample from a T-shirt, for example, when she is during her fertile period, men will say they like the smell best, certainly over menstruation or even a day before menstruation. It is maybe one of these examples, as I said, where where there is some degree of subtle advertisement of fertility. We'll talk about that again, by the way, as we talk about multisensory attractiveness and things like that in a few weeks. I guess 2 weeks. But it is an example of advertising fertility, at least to some subtle degree. So the correct answer is absolutely A in this one. Okay, great. You folks did good. All right. So we got 5 minutes, but I can use those 5 minutes to introduce you to somebody. And this person is a gentleman named Stephen Poe. There he is. Boy, I enjoyed talking to him. Steve Poe, you know, it's interesting. So what we're going to be talking about now are master sommeliers. And these are the people that are actually trained. You know what a sommelier is? A sommelier is in fancy restaurants. These are the folks that recommend wines and do the wine buying for the restaurant. They decide what wines should be purchased for the restaurant so they can then, you know, sell either bottles or glasses at the restaurant. But if you have questions about wine, not all fine restaurants have them, but many do. They will have the sommelier come out from the kitchen and help you choose a wine, and then they will serve the wine potentially. And so, you know, these are you know, people that really know wine. What a master sommelier is, though, are the people, the teachers that train the sommeliers to become wine experts. And Stephen Poe is one of these folks. Now, I have to say, I'm a beer drinker. I'm not a wine drinker. The red wine gives me headaches, so I'm probably a little allergic to the tannins or something like that. So I'm not a wine drinker. And I've had friends, though, that have been very serious wine drinkers, very serious. And I always found that, yeah, I used to go to some of these parties of my friends, and he'd, like, him and his wine drinker friends would kind of stand around and talk about wines and kind of tease the people that weren't wine drinkers, and you know, they always thought it was funny I was drinking the beer. And so my experience with wine folks, people that really, really love and are really into wine, was that you know, maybe they're going to be a little pretentious. I don't know if you ever watched the show Frasier. You know, Frasier and his brother were kind of wine snobs, that type of thing. And I was a little nervous to talk to this master sommelier, but he was so cool, and we chatted about it. I mean, you know, he was you know, he was just a regular guy, you know, camped, was in the Marines, played football. I just, you know, a very down to earth, you know, normal guy. He just loves wine and loves teaching people about wine. He just loves teaching people, you know, to appreciate wine the way he does. And I think that's just wonderful. You know, he wasn't snobby about it. He said that, you know, I told him I was a beer drinker. He said, hey, listen, I love beer too. And you know, I can appreciate how many great beers there are out there. I mean, it was no snobbiness with him at all, which was such a relief. I was really nervous that he was going to make fun of me. Terrific. But he told me about his training, and his training is really, really interesting. There are very few of these. I think actually when I wrote the book, there were like 150. The last time I looked, there were 100. Okay. Right now there's 269 in the world. I think I updated that last year. I just looked on the website, and I think there's 269, but that's in the world. It is a difficult thing to become a master sommelier. It's very difficult. And yes, they you know, they make good money. They you know, train the sommeliers. They fly around and teach people about wine and all that sort of thing. But boy, the training you have to go through, I mean, it's like, you know, being in graduate school or something like that. It is very, very serious. And he told me about his experience going through this training. What they do is they have 3 exams that they take, and it probably is going to take many years. The first 2 are in wine theory and serving decorum and things like that. So it's kind of pencil and paper sort of tests. And I think they have to demonstrate that they know how to, you know, correctly pour and store and stuff like that. But the thing that everybody gets caught on, the thing that takes multiple years to pass, is the blind tasting. And that's what I was most interested in. What they have to do for that blind tasting, the third of the 3 exams, is they have to take sips of wine that are placed in front of them and identify the wines very, very precisely. They have to identify the grape variety, okay, where the wine was grown, where the grapes were grown, and the vintage, meaning that when the wine was bottled, or what year were the grapes grown and when the wine was bottled. So they have to get very, very, very specific. And not only are they rated on how accurate they are, they have to be completely explicit about why they're choosing the wines that they're choosing, why they're saying this wine is from the Napa Valley, this wine is from a very kind of idiosyncratic area, you know, in the French Alps or something like that. They have to know this and also be able to explain why they're saying it, okay? And just very quickly, like a couple of minutes, no, 0 minutes, but they're rated on accuracy, they're rated on their reasoning and how well they're describing their experience of the wine. And they are being judged, you know, right there with 3 masters who've already passed the test to determine whether or not they become master sommeliers. And we'll talk about how this is done and how good he needed to become at this and what process he used on Thursday. Okay, see you then. Oops.